So Leo Tavares is from Penn State University, and he's gonna dovetail off of Andrew's presentation here and share some exciting work that takes a novel approach to classifying and segmenting birdsong.
All right, great. Thanks, Kalina. And thanks to the organizer. This has been great so far. So my name is Leonardo, and I'm a grad student in the physics department at Penn State. And what I'm going to present to you today is a brief summary of our attempts to develop a brain-sparse system for recognizing auditory objects. Okay. All right, so let me start off with some motivations for this. As you might have gotten from Andrew's presentation, humans have a remarkable ability to understand the speech, and we usually take that for granted, right? I mean, we have been trying to build machines to recognize speech for quite a long time now, and we very often see that it falls short from our abilities. And they're ubiquitous in the modern gadgets, but still they don't work quite as well. So there are a few challenges in automatic speech recognition that we should pay attention to. For instance, here I'm showing the performance that is usually reported for a speech recognition system is more or less on the human level, but that's on very specific data sets. And if you look closely, these data sets have hundreds of hours of training data, okay? And then if you look at the performances of these models on mismatched data, you see that they completely fail. Okay, so there's some secret sauce missing from this kind of system, and we're trying to tackle this problem. Now, speech is a little bit of a difficult problem, so we're going to switch to a simpler one, which shares some common features with speech, but that is of birdsong syllable recognition. Okay, so here I'm showing the song of Bengalese finch, data from Sakata Lab and at McGill. And now our task is to go from this to this. So an annotated version of the spectrogram where we have the start of each syllable and also the class of each syllable marked. All right, so that's our goal. Now, we're gonna do this with some constraints from the biological side. And our main motivation comes from works such as this one from Schneider and Woolley in 2013, where they recorded some units in the higher auditory cortical area from the zebrafinch, and they found that some units responded very precisely and very robustly to particular auditory features in the song. Okay, these are conspecific songs. And even more remarkable is that in the presence of a background song, these units still fire. Okay, so this is a very sparse code that is responding to specific auditory stimuli. Okay, so let me start off with what our ideas of this problem are. Here I'm showing a spectrogram that we calculate from when the sun comes in. And this is a particular syllable from the Bengalese finch. And what we're going to do is we're going to window the song. We're going to calculate many short time windows from the song, and we're going to represent them in a very high dimensional space. So here I'm showing 3 dimensions, but you should really think of this as really high dimensional, and each feature here corresponds to a single point. All right. Now that's one exemplar. We can do this for many different exemplars, and here I'm showing the same syllable, but different exemplars, so they're slightly different. But I'm taking more or less the same spectrotemporal feature for each one of them, and presumably they lie close together in this feature space. Now, other features might lie a little bit farther away, but this gives us an idea that we can maybe cluster together or train a decision boundary to capture the similar features. Okay, so that's what we're gonna do. We're gonna train these decision boundaries and combine similar features together. Okay, Now, what we do with this, here's what where I am. Suppose now that there's a song coming in. Song is represented here in the spectrogram. And now what I'm going to do is I'm going to take a slice of the spectrogram, a short time window, and I'm going to slide it on the song. Okay. This corresponds to a trajectory in this feature space. And note here that I made the trajectory go inside of the decision boundary that I just trained on purpose. And if you calculate the distance from this trajectory to the decision boundary, what you get is something like this. There's a baseline, and then it shoots up. Okay? And in fact, it shoots up above this threshold, the green threshold that I represented here, and which I defined. And that indicates to us that this particular decision boundary is capturing this feature inside of the song. Okay, so it's capturing a tiny spectrotemporal feature inside of the song. So once we've done that, then we can trace back and find the onset and offset of that detection. Okay, so that's fine. We can do that for an entire vocalization and essentially represent it as a series of decision boundaries in the feature space. Okay, and in fact, we can encode entire syllables as sequences of onsets and offsets. All right, we can do this, can carry this idea even further and construct a template bank of these onsets and offset sequences. All right, that's the training part. Now let me quickly switch to the decoding part, how we do decoding for the system. So imagine there's the same bird and it sings, and now we have a set of syllables that continuous syllables that we want to decode. What we do is we take those decision boundaries that we trained and we encode it as a set of onsets and offsets again. And now we're going to use the templates that we just trained to decode this representation. So how this is done is the following. This is just representing, again, the same set of onsets and offsets. And I'm going to take the templates. I'm going to first match the onset sequences, right? So this allows me to find the onsets in the input. Then I'm going to match the offset sequences. And that allows me to find the entire region where each syllable spans. Okay, that gives me a score function, which in fact gives me the precise start and end of each syllable. Now you may be asking, why do this? Why do the onsets and then the offsets? And the reason is really for a generalization, because imagine that we have again the same syllable E here, but now I'm going to stretch it. So the initial burst is the same, but then the harmonic stack is kind of stretched in time, right? And that means that the input onset and offset sequences will be stretched as well. So we have to be able to first capture the onset, then the offsets, and then we're going to be able to capture the entire duration of the syllable. All right, so that's the idea. Then we use the waveform to slightly correct the onsets and offsets. Now this can be a little sensitive to deletions and in the sequences. So we adopt another procedure to complete the onset and offset sequence, and that is we lower the threshold for detection. And you can think of that as kind of expanding or decreasing the receptive field of these neurons. Okay, so we tested this with 5 different Backend finches, and here I'm showing the word error rate, where the error rate is just comparing the number of misclassifications in the output string in comparison to the ground truth, right? So what this is saying is that for 10 training syllables per syllable type, which is more or less 10 seconds of recording, we get about 1% of word error rate, right? So that means that this can be quite useful in practice. Here's some examples of this idea in practice, annotating in real life some Backend finch songs. And just let me conclude really quickly. I just described a new method of recognizing auditory objects with sparse sequences. the method can be useful in practice. There's some potential application for speech recognition. In particular, we're excited about potential application in neuromorphic devices. And we're planning to release this work as an open source software for the community briefly. Okay, so with that, thanks everyone. And thanks to my advisor and my colleagues at Penn State.
Thank you very much, Leo. That was fascinating. We actually do have a moment for questions. So I'll ask the question that came up here. If others have a question for Andrew, we have about 3 minutes. So Isaac actually asks, how do you choose the appropriate time window and how does it work with speech?
Yeah, so that's a good question. For the Bengalese finch, it seemed like around 40 milliseconds worked well. But then I tried with song sparrow, and that required me to increase it a little bit. So it might depend on the specific kind of auditory stimuli. It might be a kind of trial and error thing. For speech, I would imagine that it should be a little smaller. The window should be probably smaller.
We have a moment or two if there are other questions, including for Andrew. Whether you did, there was a question about the method, wondering whether you did dimensionality reduction for the spectrogram segments. I think
that's also a good question. Mm-hmm. I played a little bit with some dimensionality reduction methods. In the end, I decided to stick with the simplest case. I'm just using a very high dimension feature space because that actually helps the training in my algorithm. But this is work in progress, so I'm not sure. Just a quick note, I'm using support vector machines, so that using higher dimensions usually works better in this case.
Great, thank you. I do actually have a question following Andrew.